What we're about to do is chapter 10. Here we go. Chapter 10. So before we start, I'll let you know, to give you a little bit of context, this is the last chapter that is going to be on the third exam, meaning when we are done with this chapter, we will have our third exam. It's going to cover the last three chapters, which is chapters 8, 9, and 10. So to remind you, chapters 8, that's all about meiosis and mitosis, cellular reproduction. Chapter 9 is the one we just finished, so it's all about genetics. And chapter 10, as you can see, is all about the structure and function of DNA. The good news is a lot of this will be review for you because in chapter 3, we talked about macromolecules, and one of those macromolecules we talked about was nucleic acids, or were nucleic acids. So you got a little bit of information, and some of this is going to be a review with just a little bit, digging a little bit deeper. So here we go. Let's jump into it. The book starts off with the guessing game. We are behind, so I'm not even going to do that. Let's just jump into it. Your book also always starts off with this. Come on. Which is the biology and society section, which tries to make what you're learning about in this chapter relevant to current day events. But your book was published before COVID-19. So the best they had at the time was a situation called Zika, the Zika virus. And it was a kind of a big deal. It didn't really hit us that much, but it was a big deal down in South America. But anyway, obviously there's a bigger deal right now, and that would be COVID-19. Long story short, what your book is trying to get at with giving you this whole biology and society thing is they're telling you this story about a virus and how deadly it can be and all the problems that the virus can solve. And then at the end of it, they say, hey, and guess what you're going to learn about in this chapter? Viruses. And that is true. So this is the chapter where you're not going to learn a lot about viruses, and you're really not going to learn a lot about vaccines. But for you to truly understand how COVID-19 works and how the vaccine works, this is the information, right? This is the ground level information. This is the foundation that you could build upon. So without knowing this stuff, you really couldn't have a good understanding of how COVID-19 works or how the vaccine works. So this is probably one of the more relevant chapters that we do because of COVID-19. So let's jump into it. As you can see, this chapter is 3 main bullet points. The first one is DNA, structure, and replication. And hey, there's some good news because structure, we talked about that in chapter 3, in the macromolecules chapter, and replication, we briefly mentioned that in chapter 8 when we talked about how it happens in the S phase of interphase. When we're done with that, we're going to actually talk about the process. How do we go? I keep saying, as you know from chapter 9, the last chapter, we keep saying that genes are the instructions on how to build proteins. So this is the chapter where you finally learn, well, how do we go from the genes, which is written in the DNA, to protein? And then, like I said earlier, at the end, we're going to talk about viruses and other non-cellular infectious agents. So any questions about what we're basically going to cover in this chapter? All right. The second word for attendance is going to be sword, like the thing you would slice and chop with, the weapon, a sword, like a cavalry sword or an NCO sword or a samurai sword. Anyway, this information I'm about to give you on the slide, let me actually put a little pin mark through it here. Your book presents this chronologically, almost like a history lesson. And I like to present it that way, but just so you know, for the exam, you're not required to remember it as a history lesson. You don't need to know these dates that I'm about to give you. You don't even necessarily need to know the chronological order of how we've discovered things. But it is neat to think about the way we discover things, because hopefully in your brain, this can be a logical process. Like, oh yeah, first we knew this. Oh yeah, and then we figured that out, and then we figured that out. So hopefully this will make sense to you, but let's talk about it. We knew by the late 1800s that DNA was known to be a part of a cell. That's all we knew by then, right? We knew that it was part of a cell. We didn't know exactly, we definitely didn't know its structure. We didn't know exactly the parts it was made up of. We certainly didn't know what we did, but we knew it was in the cells. Then we knew by the 1930s that there was one kind of molecule that was the basis for inheritance. Of course, now we know that that's DNA, right? But at the time, we didn't. At the time, and prior to the 30s, we didn't even know that there was a molecule that was responsible for inheritance. So think about all that stuff we just talked about in the last chapter about Mendel and all that stuff he figured out about inheritance. We had figured out inheritance. We'd figured out genetics and all these the probabilities and the ratios, all that, right? We had figured that out, but no one knew that there was one specific molecule that was responsible for it until the 1930s. And then separately in the 1940s, people figured out that chromosomes are made up of two components, which are DNA and protein, and you learned all about that in the last chapter. So hopefully this picture looks familiar to you, right? The DNA winds around these histones, and when that happens, you have these things called nucleosomes, and then those things even wind around each other, and then those things even wind around in a coil, right? And then that's what we have that makes up our chromosomes. By the 1950s, someone had figured out that DNA was the molecule that acts as the hereditary material. So that's a pretty big step, right? 1950s, they finally put a lot together. They knew, again, they knew that their DNA was in there. They knew that there was a certain molecule responsible for heredity, and they've put it together in the 1950s. And that is the point where they ushered in what we call molecular biology, which is the study of heredity at the molecular level. If you're taking notes, I would say, I'm going to put a question mark next to this. I'm probably not going to ask you the definition of molecular biology, but just in case, there it is. The last chapter you just learned was genetics, right? You learned about heredity in the last chapter. Now we're going to look at it at the molecular level. And again, we kind of hinted at it. The whole time we were talking about heredity in chapter 9, we were talking about alleles that are on chromosomes, right? So now we're just looking a little bit deeper. So after they figured all that out, the next effort was to figure out the actual structure of DNA. And if you remember back in chapter 1, I said that this is going to be a common theme throughout the semester, this whole relationship between structure and function. And this is a really good example of it. And you'll see why once you learn about it. But yeah, the structure of DNA was a very important thing. It wasn't just some trivial thing like, well, we just need to discover the structure just so we know more about it. No, once we knew more about the structure, then we knew more about the function. So to be clear, when I talk about the structure, what I mean is all of its atoms, like, the chemical formula, as well as how they're bonded to each other, right? All that stuff had already been identified. So again, the chemical formula, we knew there was this many nitrogens and this many oxygens, this many hydrogens, this many phosphates. We knew that, but what we didn't know was this part, the three-dimensional arrangement. So again, structure function, very important relationship there. But once we figured that out, right, we were able to figure out how DNA does what it does. And what does it do? It does these three things right here. It stores genetic information. It copies it, right? So when you go through S phase of interphase and you make a whole new cell, right? When you go through mitosis and make a perfect copy of your cells, right? You're copying your DNA. And then, of course, when you go through meiosis and you make copies of your chromosomes and they split up and you make sperm or egg, and then they come back together and you make and you make a baby. That's when you pass it from generation to generation. So again, this is the 50s we're talking about. And prior to this, we didn't know how all this happened. We didn't know how DNA did these three things because we didn't know the three-dimensional structure yet. All right. As you already know, and Rna are nucleic acids. Quite too many people missed that question on the first exam. So hopefully now you won't miss it because you're getting it again. DNA and RNA are nucleic acids. That's where the Na comes from. Deoxyribonucleic acid and ribonucleic acid. And I'll talk about those full names later. But the point here is the Na stands for nucleic acid. So please don't mess that up on the exam. Like we learned in chapter 3, we talked about polymers and monomers. So remember, a monomer is just a thing that makes up polymers, right? If you put a bunch of monomers together, you make polymers. Just like if you put a bunch of glucose together, you make starch, right? Glucose is the monomer, starch is the polymer. If you put a bunch of amino acids together, you get a protein, because amino acids are the monomer, and polypeptide, aka the protein, is the polymer. So here we're talking about DNA and RNA, and again, you need to know that they are polymers, right? DNA and RNA, those are both polymers. What are their monomers? Their monomers are nucleotides. So there might be a question on the study guide, and there might be a question on the exam that says something like, I don't know, glucose is to glycogen as nucleotides are to what, right? So glucose is a monomer of glycogen. So therefore, if I say nucleotides, therefore nucleotides is a monomer of RNA and DNA. All right. Therefore, this is the easy part. If the monomer is called nucleotide, the polymer is called polynucleotide, right? It's very easy to remember. It's one of those simple ones where the names are, that make sense, right? Protein's a little bit more complicated. Protein, you put a bunch of amino acids and you get polypeptide, right? And then here, that doesn't make sense. Here, nucleotides and polynucleotides. That should be easy to remember. Your book points out that they can be very long and have any sequence of 4 types of nucleotides. If we're talking about DNA, then the 4 types of nucleotide are A, C, T, and G. Now, if we're talking about RNA, that T is going to be a little bit different, but we'll talk about that later. And if you're taking notes, there's no need to write down the A, C, T, and G thing yet, because we're going to talk about that in more detail coming up. Right now, for this slide, what you need to know, I guess write down if you take notes, what you need to write down is that DNA and RNA are nucleic acids. The monomer is nucleotide, and the polymer is polynucleotide. Are there any questions? Okay. Here's a picture you've already seen before. This is DNA just shown in different ways. Here we see the double helix. So we see, there's that molecule of DNA and that molecule. And remember, they're all, this one molecule is covalently bonded to itself, right? That's just a bunch of that's a polynucleotide, right? That's a bunch of nucleotides covalently bonded to themselves. Same thing here. This on the right, this is a molecule. This is a polynucleotide with a bunch of nucleotides covalently bonded to themselves. And then what we have here is that these 2 strands are hydrogen bonded to each other. And I'll talk about that a little bit later, but let's go ahead and let that sink in and marinate while we talk about other stuff. But anyway, to get one particular polypeptide, this is one way of looking at it. Nucleotide, this is one way of looking at it. You've got your sugar phosphate backbone right here. Sugar phosphate. And then here you have the bases. That one happens to be a G. That one happens to be a G. Here's another base. That one happens to be a T. Here's another base. That one happens to be a C, so on and so forth. You've already seen all this in chapter 3. Then, of course, right here all the way to the right, we have the same thing, just shown a different way. The one on the middle is a very clean, easy to picture view. The one on the right is a little bit more realistic. You can see the bases and how, for example, A is a double ring and C is a single ring. And you can see the sugar here covalently bonded to the phosphate, so on and so forth. Again, this should all be a review, but are there any questions about this slide? All right, I'm going to say this again later, but now that I'm looking at it, I might as well say it now. I want to let you know that there will be no point on the exam where I ask you to identify a base by the way it looks. So I'm not going to give you this picture without the A in it and say, what base is that? You're like, oh, that looks like an A because it's green and it's double ringed. No, I'm not going to do that. So just wanted to let you know before we move forward. All right, let's talk about these nucleotides. Again, this is a review from chapter 3, so I'm going to go through it quickly. You can slow me down if you need to. But as I already mentioned, the nucleotides, so this sugar of this nucleotide is covalently bonded to the phosphate of this nucleotide, right? They are covalently bonded. And if you remember from chapter 2, covalent bonds are the strongest of the bonds. So again, it's a sugar of one base covalently bonded to the phosphate of another. And that repeats: sugar, phosphate, sugar, phosphate, sugar, phosphate. And that's where we get the name sugar phosphate backbone. So that is what keeps these nucleotides together, is the sugar phosphate backbone. You need to know that for the exam. You needed to know it on the last exam. We talked about it in chapter 3, and here it is again in chapter 10. Any questions about this slide? All right, so in your brain, again, just think about the possible questions that could come from this. The questions would probably be, how are the nucleotides bonded to each other? The first thing you need to know is that it's covalent bond, right? It's not hydrogen bond, it's not ionic bond. Each nucleotide, one nucleotide is covalently bonded to the next. And there's only 3 parts to a nucleotide, right? There's the base, there's the sugar, and there's the phosphate. So then you just need to know which is bonded to which. And if you just remember that the base has nothing to do with this, then you're good. Sugar bonded to its phosphate. Or you could even get it backwards and say a phosphate bonded to a sugar. It doesn't matter, right? As long as you know it's the sugar and the phosphate bonded together and the nitrogen base is just kind of hanging out here doing their own thing, then you're good to go. Any questions? Again, like I already said, a nucleotide is made up of 3 parts. You already know this from chapter 3 as well. The 3 parts that you need to know for the exam are nitrogenous base, sugar, and a phosphate group. For note-taking purposes, you can even just say base, sugar, and phosphate, right? Because I'm not going to try to trick you. Yeah, I mean, though, yeah, sugar and phosphate. Yes, the base is nitrogenous. Yes, technically that's a phosphate group, as you can see right here. But for studying purposes, you base sugar. Any questions? All right, you guys are really easy today. Let's nitrogenous base. First of all, like we said earlier, this is going to be a horrible drawing, but if this is the sugar phosphate backbone that I just drew there, the nitrogenous bases project out into it like this, right? They project off of it. They project off of it from the sugar phosphate backbone, right? These nitrogenous bases, they have a ring of nitrogen and carbon, and they also have various chemical groups attached. But guess what? I'm going to put a big old X through this because you don't necessarily need to know that for the exam. You just need to know, again, they project from the backbone. They are basic, right? So they're not acidic. That's where they get their name, nitrogenous base. But again, you don't need to know that. Here's a really important fact. There's 4 varieties of them. I already told you this, and I'll tell you again a little bit later. So right now you don't need to write this down. But the 4 varieties, if we're talking about DNA, are A, C, G, and T. And of course, if it's an RNA, it's not a T, it's something else, and we'll talk about that later. But again, you need to know that the base projects from the backbone, and there's 4 versions of them. As a matter of fact, this is even more important, and I mentioned this in chapter 3. The only thing that makes these bases different from each other are the varieties, the A, C, and T, and the G. It's the base that makes it different, right? It's not the sugar, it's not the phosphate. It's the base that makes it the A or the C or the G or the T. Anyway, any questions about the nitrogenous base? All right, next thing is the sugar. The sugar has 5 carbon atoms. There's 4 in the ring, 1 extending above the ring, and the ring also includes an oxygen atom. That is, of course, if we're talking about DNA, not RNA, but you know what? That doesn't matter. I'm going to put a big old X through this too. The only thing you need to know is that sugar is one of the 3 parts of a nucleotide. That's it. Yes, your book goes into these details, but I'm not going to. For the rest of this chapter and the rest of the semester, it's not important that you know these details about the sugar. So are there any questions about the sugar? All right, phosphate group. The phosphate group has a phosphorus at the center. That makes sense, right? Phosphate group. This is why nucleic acids are called nucleic acids, because of the phosphate group. That's the part that makes it acidic. And there's also a negative charge on one of the oxygen atoms. And guess what? You don't need to know any of this. This is not important for this chapter, and this is not important for the rest of the semester, so I will not ask you anything about it on the exam. I will point out that, like we talked about earlier, the fact that this phosphate group has a negative charge on the oxygen, and oxygens are so negative anyway, and double bonds are very negative as well. That's important when we talked about ATP, because remember, hopefully you recognize that phosphate group. That's part of what makes up ATP. That's right. You had the adenosine, and then you had the 3 phosphate groups. So here it is. It's back again. Those phosphate groups are pretty versatile. But anyway, are there any questions about the phosphate group? Or for that matter, the 3 parts of the nucleotides, because at this point we're done with this slide. Or DNA nucleotides are different nitrogenous base. So you could almost say that the nitrogenous base is the thing that defines the nucleotide. It is an A. It is a G. It is what makes a U, if we're talking about RNA, a U, right? That is what defines it. Like, if you remember back when we talked about polypeptides or proteins, and we said amino acids are the monomers. Remember, the amino acid had 4 parts, and every single part was the same. You all had the central carbon. They all side chain, right? That's the part that defined it. In this case, again, with the RNA and DNA nucleotides, all 3 parts are the same, other than the nitrogenous base. It is not, or defines them, if you will. Now then, your book points out that the T and the C are single ring structures, while the A and the G are double ringed structures. I'm going to put an X through this because you don't necessarily need to know this for the exam. I'm not going to ask you about this on the exam. However, this information might help you remember what goes together. I'm going to tell you a little bit later, so there's no need to write this down now, but as a little preview. A always bonds with T, or you could say that backwards. T always bonds with A, whatever. And C's and G's always go together. And one way you can remember that is the fact that it's always a single ring with a double ring, because if you had 2 double rings, it would be too wide, and if you had 2 single rings, it would be too skinny. So again, you can see thymine is a single ring, and adenine is a double ring. So if you can remember, those go together, single and a double. Cytosine is a single ring, guanine is a double ring. So again, single, double. But again, for the exam, you do not need to know which one's single ring and which one's double ring. But if for some reason that'll help you remember that A goes with T and G goes with C, then there you go. And again, at no point in the exam am I going to show you these pictures down here on the bottom right and say, all right, without the names, and say which one's which. I'm not going to ask you to define or pick out the base by the picture. Any questions about this slide? All right. The next word for attendance is going to be toilet paper. All right, so now we're going to talk really briefly about the differences between RNA and DNA. And again, this is a review from Chapter 3. You already knew this from Chapter 3. You may have even had questions about it on the first exam. I don't remember, but there certainly were questions about it on the Chapter 3 study guide. First of all, like I've already mentioned earlier, RNA does not have a T, right? There is no T for RNA. Instead, RNA has a U. So here's a quick hint, and if you're taking notes, you can write this down. If there's any questions on the exam, remember, this is multiple choice. So if there's any questions on the exam that ask you about RNA, like, which of these is the resulting RNA strand or something like that. If it's asking for RNA and it has a T in the answer, you can go ahead and eliminate that as the wrong answer, right? So for this multiple choice exam, anything that asks about RNA, if that has a T in it, then that is not one of the correct choices. Conversely, if it's asking you for about a strand of DNA, if it has a U in it, it is not the correct answer. So that is a good hint for you. That should make your testing a little bit quicker, because you can quickly eliminate probably about half of each of the problems, half of each of the options when you get to those questions. Anyway, are there any questions about that bullet point. The fact that RNA has a U, it does not have a T. All right. Another thing, and this one's a little bit less important. I'm just going to kind of give it to you once and then we'll never talk about it again. But RNA has a different sugar. So RNA is ribose, not deoxyribose, hence the name ribonucleic acid instead of deoxyribose nucleic acid. But that's it. That's the last time you're going to hear about that. For you guys, that doesn't matter. For you guys, these two sugars might as well be the same. So the fact that it has a different sugar is completely useless to understanding how DNA and RNA work at this level. But there it is. Are there any questions about this slide? All right, there's one more difference. I assume, I think it's on the next slide. It's been a while, so I don't remember. But I'm looking at this picture, and I can tell, hopefully maybe one of you can see the other difference between DNA and RNA. Does anybody know the other difference between DNA and RNA other than the U's and the sugar part by looking at this picture? How is this picture, other than the fact that it has a different sugar and it has a U, how is this picture different than all the pictures you've seen of DNA?
Is it like that was a single helix or?
I wish I could hear you, Vance. I know you said something.
I said, is it like one of them, RNA is a single helix and DNA is double?
Good, except careful with the word helix. Yes, RNA is, well, hold on. If you download the PowerPoint, you can watch this little animation that shows some of the differences between DNA and RNA. But, wait, excuse me. Oh, it's not on the slide. Let me back up. Good, I'm glad I mentioned this. And Vance, you got it. You said it correctly. Let me just write it here for you because this is important. You need to know this. RNA, this is so horrible. I hate those little lines to the side. Anyway, RNA is single-stranded, as you can see here. DNA, oh, that's so horrible. It looks like my son has better handwriting. DNA is double-stranded, and that is important, and you'll learn a little bit why. The whole fact that the whole thing about DNA being double-stranded, that's an important thing. It's not just trivial like the different sugar or even to some extent the different bases. So, but really quickly, Vance, the reason I said be careful with the word helix is because when we're talking about a helix, by definition, as you're going to learn soon, that actually is a double-stranded thing and it's twisted. So we'll come back to that. But yes, Vance, you are correct. RNA is single-stranded, DNA is double-stranded. And again, the fact that DNA is double-stranded is very important, and we'll be talking about why here shortly. So this is a 100 level class, and we're just scratching the surface, so we don't really have the time to get into this. But if you want to, you can download the PowerPoint and watch this little video about the Hershey-Chase experiment, because I'm telling you a lot about DNA and how it works and how it's structured, and I don't really tell you how we know it. So I invite you to try to figure that stuff out instead of just taking my word for it. But you don't have to. You can if you want. You can write about it for independent work if you want. Speaking of which, let's talk about at least the most important discovery, right? We're not going to talk about all the experiments, but we'll scratch the surface of the most important ones, which are, in the words of your textbook, Watson and Crick's discovery of the double helix, which is not exactly accurate. There were other people involved, and your book does mention it, but they didn't give the other people enough credit to put them on the title of this section of the chapter. Anyway, Watson and Crick, full name James Watson and Francis Crick. They are credited with determining that DNA is a double helix. So there we go. Good timing, Vance. I'm so glad you said it when you said it, because here we go on basically the next slide. What is a double helix? It is a spiral with a uniform diameter made of two polynucleotide strands. And guess what? I'm not going to ask you what a double helix is. Just know that DNA is a double helix, RNA is not. Also, I'll go ahead and put some X's to this. This is not a history class. So you don't need to know that Francis Crick and James Watson were credited with determining that DNA is a double helix. As a matter of fact, these days, there's a lot of controversy about it, and you can look it up if you want. But yeah, there's a lot of controversy. Anyway, what had happened was Watson saw an X-ray image of DNA that was produced by someone named Rosalind Franklin, and we'll talk about her a little bit later. And I hate to put an X through her name because poor woman basically had an X through her name this entire time for years since we've discovered DNA. But again, I'm not going to ask her name for the exam. Anyway, Watson saw this X-ray image. They already had some information, not just what they saw from her X-ray image. So they put it all together. They used some wire models, and then they tried to construct the double helix that conformed to all the known data about the DNA. I'm going to go ahead and go to the next slide because nothing on this slide is going to be on the exam. This is sort of just like an introduction to what we are about to talk about. There's some pictures. There's old Watson and Crick on the left, pretending like they're doing work. Seriously, I'm going to share some documentaries with you guys. You should watch them. Obviously, they'll be for extra credits, so you can watch them and answer the questions. But these two people were pretty interesting. They were basically slackers. They weren't like the best students, yet they were credited with one of the biggest discoveries in biology. Anyway, there's Rosalind Franklin on the right. That's what they look like. As you already know, this is sort of like a review from chapter 3 as well. The model of DNA that they made was like a ladder that was twisted into a spiral. So I don't need to explain that to you. You guys have all seen the pictures of DNA. It does look like a ladder that somebody had twisted into the spiral. The sides right here, of course, this one's not twisted into a spiral, but you can see this picture down here, the ladder. These sides right here, that represents the sugar phosphate backbones. Wow, that's mind-blowing, right? And the rungs right here, those things represent the bases. They're connected by hydrogen bonds. Wow, mind-blowing, right? We've said it again. We're saying it again and again and again. You guys already know this. And as you already know, it is the base pairing rules that dictate the combination of bases that forms the rungs. What we're saying there is if this one has a T or an A, then this one has to be a T, right? Those are the rules. Them's the rules. We can't break them. If this one's a C, then by the rules of base pairing, this one has to be a G. That's all we're saying. So again, nothing on this slide is new information, but are there any questions? Okay. Next one. Next slide. Here we go. There's a ladder, and it's been twisted. Mind-blowing, right? That is the model of DNA. That should look very familiar. Here's some more pictures of DNA. Wow, look at that. It's like you've seen this before, right? So again, the important things to point out, a molecule of DNA is two strands, right? It's a two-stranded thing. Each strand is covalently bonded, a sugar to a phosphate, to a sugar, to a phosphate, to a blah, 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 right? Each strand is covalently bonded to itself. This strand is covalently bonded to itself. However, this strand, this is the same strands I'm talking about, but it's easier to visualize this with this middle picture. These two strands are hydrogen bonded to each other, which is very important. Think about this. The thing that keeps an individual strand together, I'm just going to call this strand A. The thing that's keeping strand A together is a bunch of covalent bonds, the strongest of the bonds. The thing that's keeping this strand together, the strand B is what I'm going to call it, a bunch of covalent bonds, the strongest of the bonds. The thing keeping strand A together with strand B is this, hydrogen bonds, the weakest of the bonds. So keep that in the back of your mind as we go forward. The thing that is holding an individual strand that will be relevant here in a second. Another PowerPoint. If you download the PowerPoint, you can watch this little animation about the DNA double helix. In 1953, Watson and Crick published a paper proposing the molecular model for DNA. And again, this is not a history class, which is why I put an X to this. I'm just teaching it to you as we learned it, as humans discovered this, but you don't necessarily need to know either the dates that these things happened or how they happened chronologically. But yeah, this is when it happened. They figured it out. They proposed a paper. Actually, if you download this PowerPoint and you click this picture of the Nobel Prize, then it'll show you a quick documentary about how that happened. Anyway, in 1962, Watson, Crick, and Wilkins, and I didn't even talk about Wilkins. You can look him up. He's just a third person that was involved in this whole situation. They received a Nobel Prize for doing what they did and published in 1953. And I like that your book points out that Rosalind Franklin, she deserved a share, but she was already dead by the time that they were awarded the prize. and the Nobel Prizes are never granted posthumously. So anyway, just a big old exodus through this. It was in your book, so I included it. The point here is they figured out the structure of DNA, and obviously it wasn't trivial because they won a Nobel Prize for it. So let's talk about why it wasn't trivial. Again, Watson and Crick wrote in that paper that they published that immediately they wrote that the structure of DNA immediately suggests a possible copying mechanism for the genetic material. So remember, at this point, not only did nobody know the structure, because that's what they figured out, but also no one knew exactly how it worked. How was it copied? How did you go, like, how did parents pass down their DNA from one, from them to their offspring? How did that happen? No one had any idea. And it turns out it was the structure of DNA that explains it. And let's talk about that. Let's talk about DNA replication. We've got about 6 minutes left, 6 minutes left. We can finish this part. DNA replication. As you already know, where's my pointer here? Come on. There we go. As you already know from chapter 8, when a cell reproduces, it has to duplicate its information, right? It has to provide chromosomes, specifically, that's the information we're talking about. It has to provide a copy to the new offspring, but also keep a copy for itself, right? We learned that in chapter 8. Basically, what we're talking about in the big picture there is mitosis. But if we're talking about the copying part, then we're specifically talking about S phase of interphase. But anyway, you already knew that. And what the new information here, again, is what Watson and Crick figured out, because they figured out the structure. And again, it's not a trivial thing. It's not just like, oh, cool, you figured out what it looks like. No, it's bigger than that. The structure let us figure out how things work. And this is what I'm talking about. So again, each individual strand, right? So let's say strand A here and strand B. Strand A is held together with covalent bonds. Strand B is held together with covalent bonds. But the thing that is holding strand A to strand B together is the weakest of the bonds, which are hydrogen bonds. So think about that. If you were to grab, all right, let's put a line through this. Forget that the bottom half of this picture exists. And just look at this DNA up here. If you were to grab this strand of DNA on the left and pull it, and if you were to grab this strand of DNA on the right and pull it the other way, what's going to happen is they're going to open up, right? It's not like this strand A is going to rip apart or strand B is going to rip apart. No, strand A is going to be separated from strand B, because here the thing that's keeping them together is the weakest of the bonds, which is hydrogen bonds. So as soon as they saw this, and think about this, now we can look at this lower part, and they also knew that once you pulled that thing apart, all of a sudden the thing that defines the base, the thing that defines the nucleotide, which is the base, right? There's a G, there's a C, there's an A. As soon as you pull that thing apart, which is relatively easy to do, then all of a sudden you have a code, right? Or the code's been there, but now all of a sudden it's exposed. And that's how they figured it out. They said, all right, well, this must be a template. All you have to do is open this DNA up and then just bring in the new nucleotide based off of the code. If there's an A, then we're going to bring in a T. If here's a C, we're going to bring in a G. If here's a G, we're going to bring in a C. So again, this was a revolutionary thing to figure out how it worked, just by figuring out the structure. If you download the PowerPoint, you can watch this. This is a really neat animation because it is very accurate as to what it looks like. There's a cellular machine that we're going to talk about later that actually clamps on to the DNA and it rips it open, right? It rips those two those two strands apart so they can read the code and bring in new nucleotides and make new DNA. So everything we talked about in S phase of interphase, that's what you can see in that little video. Same here. This video shows the same thing, except it's really basic looking. So this one's easier to follow because it's not what it actually looks like. But the last one is more of what it actually looks like. Same thing here. This one's more of what it actually looks like. If you watch this and you see words that you don't recognize because they're not in your textbook, then you can ignore them. Like primase is a word that we don't talk about, for example. All right. Actually, let me back up one more step. Actually, that'd be a good place to end it. One thing I didn't mention on this slide, because this is a question that is on your study guide and a question that will be on your exam. I think the question says something like, a new strand or a new double helix of DNA consists of what? A new strand of DNA consists of what? So first of all, let's really quickly define what we're looking at in this picture. I think I've erased enough. What we have here up top is the old strand, right? This is the old double helix. Old double helix. And what we have here at the bottom, remember, this is a copying mechanism. Remember back in chapter 8 when we talked about S phase, we had one chromosome and then it duplicates into two sister chromatids, right? So what we have here is two new DNA double helixes, right? There's a new one and there's the other new one. And the question on the study guide and the question on the exam is going to say something like, a new double helix of DNA consists of what? And one of the options is going to say two old strands of DNA. And old here is represented in blue, the strands anyway, right? And that's obviously not the case, right? And then the other option is going to say two new strands, and the new here is the gold, and that's not the case. What makes up a new DNA double helix? Well, we have one old strand, which is the blue, and one new strand, which is the gold. That is a test question, and it should make sense, right? So every time you copy DNA, the old strand is split up, right? And one half goes each way. And then from the template of the old strand, you build a new strand. So again, each new double helix gets one old strand and one new strand. So the last word for attendance is going to be fire pit. And when we come back, there we go. When we come back on Monday, we will talk about that cellular machinery that makes this possible, that makes the DNA replication possible. So are there any questions while I'm still recording?
What was that last word again?
Fire pit was the last word. Like a place that you would burn firewood in your backyard. Fire pit. Good question. Anybody else? All right. In that case, I will stop presenting. I will stop recording. Again, I will